Hi folks, this is a short video to explain the results of your protein digesting experiments. So all the test tubes are labeled here. Here's P1. To test tube P1 you added albumin and distilled water. And then after incubating at 37 degrees Celsius for an hour, you tested for the presence of peptide bonds, essentially. So the reagent you used to test that was called Biuret's reagent, and if there are more peptide bonds, the solution will appear violet. So if you have more peptide bonds, that means you have a longer protein. Let me actually draw this out. If you could bring the video camera up here, just so this makes sense to you all. So let's say these are all amino acids. Joining the amino acids are bonds called peptide bonds. This could be quite a long chain of amino acids. So Biuret's reagent will react with all these peptide bonds and give off a violet color. So this is a longer polypeptide, which would be kind of like albumin. It's actually quite a bit longer than what I've drawn out, but I think you get the idea. So if protein digestion occurs, that means that an enzyme, a peptidase, breaks some of these bonds apart. So then you'll end up with shorter oligopeptides. Okay. So if you have shorter oligopeptides, you're going to have fewer peptide bonds overall, because some of these bonds have been broken. So then Biuret's reagent has fewer peptide bonds to react with, and thus digested protein appears pink in response to Biuret's. Okay, so with that background knowledge, let's take a look at our things here again. Should I flip this around, or?
It's okay.
Okay. Alright, so test tube A1, we just have albumin and distilled water, and this looks violet. So there's a lot of peptide bonds in the protein albumin. Here's test tube P2. In this test tube we had albumin and hydrochloric acid and distilled water, and mixed that with Biuret's reagent, and it still looks violet, so that indicates that protein digestion has not occurred. In test tube P3 we have albumin, pepsin, and distilled water. And pepsin, remember, is secreted by the stomach, and its job is to digest proteins. And this test tube looks kind of pink, right? See the difference between this and that? It's kind of slight, but I think it looks pink. You see the pink on there?
It's kind of hard to tell.
Slightly. Well, maybe it'll look better in lab, but there is a little difference between that and these two. This is pink. But test tube P4 looks really pink, really obviously pink. In test tube P4, there was albumin, the protein, hydrochloric acid, and pepsin. So pepsin is a protease. Pepsin breaks peptide bonds. But remember, its optimal pH is around 2, which is a very acidic pH. Hydrochloric acid is a very strong acid. So the hydrochloric acid you added made that pH optimal for the action of pepsin. And when you incubated this test tube for an hour at 37 degrees Celsius, a lot of protein digestion occurred, as is indicated by this pink color. So you ended up with very short oligopeptides, maybe some dipeptides, maybe some free amino acids as a result of pepsin at that high pH. Excuse me, at that low pH. All right, that's all I have to say. Thank you very much.